My name is Raghu Ramanathan, and I work for Pfizer as a bioanalysis group leader in Groton, Connecticut. And I lead a team of scientists who are involved in measuring and quantitating drugs and metabolites in animal and human samples so that we can bring safe and efficacious drug to humans faster and with a costly manner. Drug-drug interactions, naturally, if you're taking only one drug, drugs are metabolized by enzymes in liver and other organs and then eliminated from the system. When you take more than one drug, it is possible one drug can enhance or limit the potential of the liver enzymes or the gut enzymes in metabolizing these drugs. Or if you take, for example, drug A and drug B could inhibit drug A's elimination. So this is why it is important to understand drug-drug interaction, and so that we can adjust the dosing as needed so that we don't have any toxicity or any ineffective drug when we're taking combination of drugs. So the established method currently is based on in vitro or cell-based assays projecting DDI potential of drugs, and then that is extended into the clinic. So one of the difficulty is that DDI projections based on cell-based assays provide higher incidence of false positive, almost like 30% false positive. So is there a way for us to find out how we can implement other options to get the DDI more reliably so that we can limit dosing of probe drugs for evaluating DDI in the clinic? So the challenge is that we have to find out when to dose these drugs in the clinic, and difficulty with that is that unnecessary dosing of probe drugs. For example, for statins, if we are dosing trostatin unnecessarily, then that is going to prolong the clinical trials. So the idea is that can we use endogenous biomarkers that already exist in the system as a surrogate marker for understanding the drug-drug interaction of transporters and some of the CYP enzymes. The transporter sciences and CYP enzyme sciences are rapidly evolving and always looking for new technologies to boost our understanding of these enzyme-based drug-drug interactions. So the focused issue that is coming out in May timeframe is going to focus on new technology in mass spec, as well as it is going to propose several endogenous biomarkers for evaluating DDI early in the clinical trials so that we don't have to continue DDI studies into late stage clinical studies. So from the get-go, what we want to do is we want to support or refute what is being projected based on cell-based assays. We want to take advantage of endogenous biomarkers such as copoperin 1, copoperin 3, bile acids. Both are indicating whether we have organic anion transporting polypeptide liabilities. And that will tell us if there is a DDI is possibility, and then we can stop additional clinical studies for drug-drug interactions, or we can bring in new studies for drug-drug interactions. Similarly, the special issue will talk about techniques for multiplexing, getting more information, more than one biomarker using high-resolution mass spectrometry, and a couple of other techniques, including sample pooling approaches, as well as using high-resolution mass spectrometry for multiplexing for in vitro biomarkers. Yes, in the special issue, we discuss techniques involving, again, high-resolution mass spectrometry. Use of high-resolution mass spectrometry for quantitation is still evolving. Typically, we use triple quadrupole-based assays for quantitation. By using high-resolution mass spectrometry, we can multiplex, meaning that we can get more than one biomarker information from a single assay. So that is really highlighted in this special issue. And also we have highlighted some of the capabilities around microflow techniques. That hasn't been really fully discussed in the issue, but that is something that we are looking at as a possibility for future applications.